Hello, students. Can you hear me? Are we live? Yes, we are live. Hello, how are you all? Can you hear me? All of you can clearly hear me and see me? Let me know quickly in the chat session. Yes. How are you all doing? How are you all doing? Hello, hi. Yes. Yes, let me know quickly if everything is clear. Can you see me? Can you hear me? And you can also see my slides? Yes? Okay. Yes. Okay. You people have picked up on my namaste, no? So I'm going to say namaste, okay? Because all of you have been telling me on my special class as well as on YouTube, right? You people have picked up my shorts where I'm telling that is students, that is not namaste, it is namaskaram, okay? So yes, namaskaram. So welcome you all back. So students, quickly share the link with your friends. Like I always tell you, more the people, more the fun, right? So quickly share the link with your friends so that more students can join and we can understand one very, complicated concept in a very simple way. Okay. So students, understand this. In this chapter, I am not going to rush in completing the concepts. Okay, we are going to understand each concepts and then move on. Okay, so the videos are going to be short, sweet, but very, very helpful. Okay, very helpful. So whenever, like when you're preparing for your NEET 2024 later, it will be really helpful that, okay, this concept is here. So we will have a series of concepts. Okay, students. So the videos are not going to be long and anything. It is going to be really short, sweet. Okay? Yes. So those who are new to this channel, welcome to Unacademy NEET English channel. And if you do not know me, my name is Gopika and I'm your biology educator. Okay? And students, what is the first thing that I say you all the time? Right? Maintain the energy throughout the class because the concepts in photosynthesis in higher plants is a little difficult. So I do not want any of you to lose motivation and quickly share the link with your friends. Okay. I have also shared the link on Telegram. So those who didn't see the link, yes. Okay. Fine. Let's start the class. Before I start the class, okay, I have told you about this. Now, students, understand it is really important to understand where do you stand, right? Whenever, see, why do we conduct exams? Generally, we conduct exams so that we know where we stand, right? So, like that, to understand where you stand, okay, where you stand, it is important for you to write exams or attend exams, especially when it is all India exams. You don't have to worry because the way the corrections are going to be, it's going to be in a very amazing manner. So you will know what is the fault that I have done, why is this going wrong. As I always tell you, NEET 2024, the competition is going to be really tough and the students number also is going to increase because you know that there was a reduction in chapters. Okay, so quickly, link is in the description box. Quickly go and register for unsat, and we know that Unacademy brings it in the best way possible, right? Yes. And students, we also have crash courses which are opening for you. Okay, you can look at the prices here. The prices are very, very convenient, right? Only batches is 9,999, and then we have 6 plus 2 months, and then we have 6 plus 2 iconic. So you have plus classes as well as iconic classes. Okay. Yes. Oh, yes, all my future doctors have to gear up and prepare, right? Like I always call you people future doctors because I know that you people are capable, just that you need a little push and motivation, and that we are ready to give you. Only thing you have to do is go click in the description box. The link is there. Just join our batches. Okay. Yes. And students, we have spoke about this Unacademy All India Test Series. Okay. So that is preferably for students who are class 12 and droppers, and for students who are in class 11 online can also join. Okay, fine. Then Yes, what and all will you get? What is the benefit of joining this? Students, you're going to get notes, you're going to get mind maps, you're going to get flashcards, you're going to even get test series. You're also going to get evaluation. Where do you stand? Okay, so do not hesitate. Quickly join. The description is right below, and the only thing that you have to do is put my code, right? Use my code. Go pick up and join quickly. Okay, join quickly. Yes. Okay, so let's start the class. Before I start the class, let's quickly revise what we did last class. Okay, so students, why am I doing the revision is because if you have to understand this concept, you have to also watch my previous video. If you haven't seen cyclic and non-cyclic photophosphorylation, I'm sorry to say this is going to be really difficult for you to understand. Okay, chemiosmotic hypothesis or your chemiosmotic theory. What does it mean? Okay, so when I explained you non-cyclic and cyclic photophosphorylation, I used a statement telling that when this process is happening, right? When this process was happening, I told you people that the your ATP synthesis is happening, right? I told that ATP is getting synthesized. Correct? ATP is getting synthesized. Now, I didn't tell you exactly how ATP is getting synthesized. I only told you people that in the CY, that is your cytochrome complex, ATP will be synthesized. How? When ADP adds with your phosphate group. This is what I told you. Now, students understand. For your ATP synthesis to happen, we have a whole other hypothesis. And that hypothesis is called your chemiosmotic theory. How many of you understood this? How many of you understood this? Yes, tell me quickly. How many of you understood what is chemiosmotic theory? Yes, all of you understood. That means what? Whatever we studied in your non-cyclic and cyclic photophosphorylation, I used a term that the CY, that is your cytochrome complex, is forming the ATP with the extra energy, right? But we couldn't add this whole synthesis in that cycle. That is why we are studying it as a separate. We are studying it as a separate hypothesis by itself. Okay? Yes. So let's try to understand where did this come from, what did we know, who invented it and all that. Okay. So students, chemiosmotic theory or theory describes how energy molecules, that especially your ATP, adenosine triphosphate, is synthesized. Okay. Now, it was put out by a British biochemist named Peter Dennis Mitchen in 1961. Okay, he was the person who understood that maybe ATP is synthesized by the extra energy, but there should be some gradient that is going to happen, or there might be some complex that is going to do the job, right? So that was done by your Peter Dennis Mitchen. Okay, in plants, during the light reaction, photosystem help chlorophyll to absorb light. Very, very important. I told you when we spoke about photosystems, I told you it is like your hero and your friends, right? So what is going to happen? Hero also is going to have some energy. The friends also is going to have some energy. But all of them, when all of them hit together, is the effect double or single? Tell me. Like I told you last time also, if teachers are helping you to crack NEET, is the energy double or single? Tell me quickly in the chat box. I'm waiting for your reply. Yeah, no spam, no spam. Double, right? See, now when you people are studying with me, or say when you people are studying with all of us, our Unacademy NEET English channel teachers, the energy is double, right? The Josh is double. Okay, and especially when you see all 5 of us, the energy is going to be triple, right? Same way, students, what is going to happen? Chlorophyll A by itself, that means you by yourself, is a capable candidate, okay, for NEET 2024. But when we accessory pigments come in close with you, what is going to happen? The NEET 2024 results is going to be double happy thing, right? So same way, what is going to happen? Chlorophyll A is there, it is going to absorb some sunlight. Accessory pigments also are also going to absorb some sunlight. Now all of this is going to emit or give out a pair of electrons. That is what we saw in non-cyclic. Now, during that whole process, we saw that when this electron was transported through the electron transport chain, it reached a point called as cytochrome complex. Okay, I'm telling for students who do not have any idea and has joined the class first time. Okay, so your challenge for today is to go back and watch cyclic and non-cyclic. Otherwise, this concept is going to be really, really difficult. Yes. So what are they trying to say? As a result of the absorption of light, we also had an electron that was emitted. Now, during that process, because the electron went out and it did not come back to photosystem, we had to do photolysis of water. So, hydrolysis of water takes place and releases electrons and protons. Okay. Now, the electrons get excited to higher energy level and are transported by the electron transport chain. Okay. Now, look here. This is the important point. This creates a proton gradient. Okay. This creates a proton gradient, which I will be explaining it to you people. Do not get worried. Just understand proton gradient. Okay, understand the term, right? Yes. Some protons are used by the photosystem 1 for reduction to NADPH. Do you all remember? All of you tell me quickly in the chat box which is the enzyme to convert NADP to NADPH. Only if that is there, that whole process will happen. Let me know quickly. Quickly let me know in the chat box which is the enzyme that is going to help for the formation of NADPH. Yes, very quick. No, yes. David got it wrong. FD is your ferredoxin. Ferredoxin is your electron carrier, right? After that, it is going to one enzyme is doing a job. Yes, very good. Very good. NADP reductase. NADP reductase. Do not get confused. It is not synthetase. It is your reductase. Okay. Even though NADP synthesis is happening, we use reductase. Okay. Because addition of a hydrogen is happening, right? So, when the proton gradient is collapsed, it releases energy and the protons are carried out back to stroma through F0. F0, do not worry. I will explain you. But what you have to understand is that we used a proton for forming NADPH2 in our that is your non-cyclic photophosphorylation. Now, what all happened? What all happened? Students, are you all with me? Yes? Yes? No? Tell me quickly. Yes? Okay. Yes. Where is the Josh here? No likes in the session. No likes at all. So many of you are watching, but no likes. Yes. Okay. So students, we will try to understand what were they trying to say. Now, I will explain you with a diagram because that makes more understanding, but just understand what this is all about. Okay, students, your chemiosmotic theory is basically synthesis of your ATP. Synthesis of your ATP. How is synthesis of ATP happening? When your ADP gets added with a phosphate and forms an ATP. This process, right? This process took place in your cytochrome complex. Cytochrome complex. Right? So much is clear. Okay, it took place in your CY, right? Cytochrome complex. Now, very, very important thing that you people need to understand is in your non-cyclic, okay, non-cyclic photophosphor. Are you able to see? Yes. Rylation. Okay. During your photophosphorylation. 1 minute. Yes. During a photophosphorylation, we saw that, okay, photophosphorylation, we saw that the electron was in so much energy that it was continuously moving from one point to another point, right? Yes. Now, when this electron was moving, we also noticed one thing, that the electron that was released did not come back to our photosystem 2. Okay, it did not come back to our photosystem 2. Right? It did not come back to our photosystem 2. That is why we call this reaction as non-cyclic photophosphorylation. Now, what you have to understand is that in your chemiosmotic hypothesis, we have some protons, we have electrons, and we have a set of reactions that is happening in the thylakoids, that is happening in the inner membrane and in the outer membrane. Okay. Now we are going to understand that. Students, now tell me one thing. Now your thylakoids, right? Let me draw your one thylakoid. has a double membrane. Okay, I'm drawing a lot of space between so we can explain it better. Okay, look here, students. What and all is happening? What and all is happening? What is happening here? So I'm going to say that here, okay, here we have NADP reductase, right? NADP reductase. Yes. So here we have NADP reductase. Yes, NADP reductase is there. Correct? Now, here, okay, here you have your splitting. Where does splitting of water take place, students? Where does splitting of water takes place? Is it in the outer membrane or is it in the inner membrane? Last class I told you all this, so this is like a revision. This is like a revision. Tell me, where is it happening? Where is it happening? Is it happening inside or outside? Is it in the outer membrane or is it in the inner membrane? Okay, I'll write this down. Yes, whoever tells the answer first has revised it. Yes, very good. It happens in the inner membrane. So what is going to happen? A splitting. So I'll draw here. Okay, splitting of water is taking place here, right? Splitting of H2O, right? Yes, splitting of H2O is happening. Now, these three locations always keep in mind because I'll tell you why these locations are important. Okay, so NADP reductase is in the outer membrane, splitting of water is happening in the inner membrane. Okay, and we have one more thing. What is that? One more thing is there. This is your stroma. Okay, this is your stroma. Your stroma also is going to do some job. Okay, I'll tell you what your stroma is going to do. Okay, let's see. So students, this is what is given in your NCERT. So I've used your NCERT diagrams because I've seen a lot of students telling, ma'am, do not take anything other than NCERT. Okay, so we're going to understand from your NCERT. I want all of you to focus here. The diagram might look complicated, but it's a very simple diagram. Okay, now look here. Here, this is your outer membrane of a thylakoid. This is your inner membrane of the thylakoid. Okay, now look here, we have our photosystem 2. Now what you have to understand is, this is your photosystem 2, right? Photosystem 2. From your photosystem 2, electron is going, right? Electron is going and it is going to reach your PEA, that is your primary electron acceptor. From there, the electron is going to come through your electron transport chain. Same way, you're going to keep this in your mind. And see, look here, you have your PS system 2. Okay, now electron is going to go. Okay, it is going to go. Can you see, all of you see here PQ? Can all of you see here PQ? Plastoquinone? Yes or no? Yes or no? Can all of you see plastoquinone? Students, you can open your NCERT. You can open your NCERT and look if you're not able to see what is written in the pink. Okay, because there it is it is written in here. It is written black here, so it might not be visible on screen. So open your NCERT, all of you. All of you open your NCERT and try to look and try to understand. Okay, I will tell you what is happening. Okay, so what is happening here? You are only going to see what I'm going to say. Okay, look here, we have our PS system 2. From the PS system 2, the electron is going to escape, right? Then who is going to accept it? Primary electron acceptor. From the primary electron acceptor, it is going to come to your plastoquinone, correct? From your plastoquinone, it is going to come to your cytochrome complex, that is your CY. Okay, now they have enlarged your cytochrome Y because your cytochrome Y is where your ATP got synthesized. Do you all agree on that? Do you all agree on that? Remember when we did non-cyclic photophosphorylation, I just revised also. Cytochrome is the one where ATP synthesis is happening. That is why they have enlarged it. Yes, very good. Now, I want all of you to respond, okay? All of you to respond. The Josh is very less and the likes also are very less. Quickly like the session. Quickly like the session. Quick. Where is the Josh? Where is the Josh, students? If Josh is not there, then the energy to teach also is very less. Okay. That was such a complex topic also. So I need more energy in the class. More energy. Yes. More energy, more likes. Okay. Likes have to increase. Quick. I'm going to see my future doctor's energy. Yes. Great. Fine. Okay. So here, what happened? your PS system 2, the electron has come to PQ. From PQ, it is going to cytochrome. Now, what is going to happen? From your cytochrome, we understood that from your cytochrome, it went to PC, that is your plastocyanin. From plastocyanin, we went to PS1. So, understood this? What is all these three things? From PS2, it went to cytochrome, right? From cytochrome to PS1. Now, all this is present on your outer membranes, right? It's all present in between, right? The outer membrane and the inner membrane. Students, cycle is clear. Cycle is clear. Can you see the connections they have drawn here? Now, what is exactly happening? Now, I told you that your cytochrome uses the extra energy to convert ADP to ATP. Now, students, not only that, your cytochrome is going to do one more thing. When the energy is passed like this, right, it is going to accept a proton. It is going to accept a proton from the stroma. Now, see, look here, all of you. Stroma. This is very, very important. Okay. The stroma is the outside one. This is your thylakoid. So, what is going to happen? The proton from the stroma is accepted by the cytochrome. So, it becomes a proton carrier. Okay. So, this becomes the cytochrome. Can I call it a proton carrier? Yes or no? Yes or no? All of you let me know if it became a proton carrier. Yes? Yes. Okay. So, it became a proton carrier. Now, understand one thing. Sorry, 1 minute. Yes. Now, understand one thing. This proton carrier, now the cytochrome has accepted a proton. Now, this proton will go into the lumen. This proton will go into the lumen. So, can you see here? Can you see here? They have drawn this H plus, right? So, students, this proton is carried by the cytochrome and it is getting into the lumen. Now, if you ask me why, ma'am, why is this happening or something? Students, understand the cytochrome is actually more of an electron transporter, but it is mainly a proton carrier. That means that is the job of cytochrome complex. Okay, that is why last class I remember one of you have asked me a doubt. I think it was in the special class. During a special class, one student asked me, ma'am, they have not given the option, okay, but they have given cytochrome as an option for proton carrier. Students, so understand cytochrome mainly does proton carrier job, but during the process, it also transports electron to your plastocyanin. Okay, plastocyanin. All of you are clear on this? Once you understand non-cyclic photophosphorylation, this is very easy. Okay. Now, what has happened? The proton has come. The cytochrome accepted it. It automatically went into the lumen. Not very difficult to understand, right? It passed into the lumen. Now, in the lumen, one more process is happening for formation of protons. Can any of you tell me what is that process? What is that one more process that is happening for this proton, for getting our proton? That proton we use to convert NADP to NADPH2. Who is going to tell me? Who is going to tell me? Yes, very good, very good. Okay. Yes, I cannot read your names. Okay, but then yes, good job. Splitting of H2O, right? Splitting of H2O is happening. So look here, all of you look here. There is going to be a complex that will help for photolysis of water. Now this water has got split. What and all are we getting when we split water? Let's write it here. What and all are we getting when we split water? Here, let's write it here. We will get H2O gets split, right? Gets split into 2H plus 2 electrons plus your nascent oxygen or 1 molecule of oxygen or half oxygen, right? This is what we are going to get. So students, now what has happened? The cytochrome also accepted protons. This oxidation of or your splitting of water also has given some protons. Now tell me, in the lumen, will the proton concentration be more or less? Will the proton concentration in the lumen be more or less? Quickly let me know. Will it be more or less? The lumen, more. Very good, more. Right? That means what? Now we have got protons from cytochrome. We have also got protons from our splitting of water. So here, this region, right? This region in the lumen is going to have high concentration of protons. So here, this is going to be, so this place, okay, I'll write it outside then. This place is going to have accumulation of protons. Students, where is it going to have? The lumen is going to have accumulation of your protons. Accumulation of your protons. Got it? Now, tell me, normally if diffusion has to happen, that means the protons have to move from a region of higher concentration to a region of lower concentration. Agreed or not? So, it is going to move from where to where? It is going to move from the lumen. Okay, it is going to move from the lumen to the stroma again. Okay, it is going to move from the lumen to the stroma. Okay, how many of you got this? How many of you understood so far? Let me know quickly. Let me know. Okay, so let's write here. Let's write here. Yes, okay, let's write H2O gets split into 2H plus 2 electrons plus nascent oxygen, right? After that, what happened? Because splitting of this happened, there was an accumulation, okay, accumulation of protons. Where accumulation of protons was there? In the lumen. Now the protons are going to move from a region of higher concentration to a region of lower concentration, that is to your stroma. Now students, outside is the stroma. The white color region that you're seeing is the stroma. Yes? All of you got it? Yes, very good. Lumen to stroma. Now what is going to happen? Will the proton just move like that? No, the proton will not move like that. For that we have to have one proton gradient, or say some particles that will help for the proton to move from the lumen to the stroma. So here, all of you look here. All of you look into your NCERT. Can you see a green color box? Can you see here a green color box which is labeled as CF0? Okay, students, so understand, we are going to have a proton gradient, okay, a proton gradient or a proton carrier, that is called your F0 and your F1. Okay, that is what here they have given. F1 is also, it behaves like an enzyme, and that enzyme is called ATP you can tell ATPase, most easy, ATPase. Okay, in your textbook it is given, no? ATP synthesize, it is given, right? So there is a channel for this proton to move from the lumen to the stroma, that is from the higher concentration to a lower concentration by the help of diffusion. Okay, now what you people need to understand here, what is going to happen? The proton is going to move, right? The proton is going to move. Yeah, the proton is going to move from your stroma, right, like this. It will reach here. Now what is ATPase supposed to do? Now I told you your F1 is going to act like an ATPase. That means it is going to synthesize ATP. So what is going to happen? This proton also is coming with a lot of energy. So that energy will be used up by your F1, that is your ATPase, to convert your ADP, right, to convert your ADP to your ATP. Okay, to convert your ADP to your ATP. Okay, so this is why we call this as an ATPase. Okay, ATPase. Okay, now students, now what has happened? From a region of higher concentration, that is from your lumen, it has come to stroma, right? From lumen it has come to stroma. Now one more job is done, like, you know, proton has to do one more job. What is that job that proton has to do? What is that job proton has to do? That is to convert your NADP to NADPH, right? We have to synthesize NADP, right? Because this is a light reaction. This is a non-cyclic photophosphorylation. That means we have to synthesize NADP. Yes? Yes. Okay, great. Now, so that what is going to happen? The proton that will be coming out, can you see an H plus here? That proton will be used up by your NADP to get converted to NADPH. NADPH. How many of you understood the reaction? How many of you understood the reaction? Okay. So what is going to happen? Your NADP, okay, write it here. NADP will get converted to NADPH2. Okay, NADPH2. All of you got it? I want quick reaction. Tell me quickly if you all understood it. Yes, very good. Very good. All of you? Great. Okay. Yes, same. ATP is ATP synthesis, same. That means we synthesize ATP. So you can tell it there. Okay, you can tell it that way. Got it? Yes? Good job. Okay. Now, students, let's try to understand what and all we understood from this, right? What and all did we understand? Okay. Now, you all are going to answer me. Okay. You are all going to answer me. I'm only going to write what you people answer. Okay. Can we have a question round? Quickly let me know. Can we have a question round? Yes? Yes. Okay. Yes. Okay. So chemoosmotic hypothesis, right? Chemoosmotic hypothesis. What and all has happened? What and all has happened? Okay. So this process mainly does, what does it do? It does synthesis of ATP. Okay. This is a revision for all of you. Synthesize for ATP. Now, not only does it synthesize for ATP, but it also helps in synthesis of NADP, right? It also helps in synthesis of NADP. Are you all there? Yes? Yes? Great. Okay. Now, what is going to happen? Okay. Gaurav, students, all of you, please tell hi to Gaurav. He's been telling hi to each one of you. All of you tell hi quickly so Gaurav can concentrate on the class. Yes. Okay. So, we have to synthesize NADPH2, right? NADPH2. Now, what else are we going to do? So now here, what are the important things? Okay, let's first understand what are the important things or what are the sites, right? Let's understand what are the sites. All of you? Yes? Okay, so what are the important sites? I'm going to ask you people question. I want quick answers. Okay, tell me first. Site of water splitting. Where does site of water splitting happen? Site of water splitting. All right, H2O. Site of water splitting. Very good. Yes. Where does it happen? In the inner side of the membrane. Why I'm trying to make you understand this is because it is, if you do not understand this, right? If you do not understand this, you're not going to understand what is happening. From where is proton coming? From where is all this happening? You will not understand. Okay. Yes. Okay. So next, this answer is what? Inner membrane. Inner membrane. Yes. Very good. Now, next question to all of you. Where is NADP reductase present? Where is NADP? Okay. NADP reductase present. Where is NADP reductase present? Quickly. Yes. Outer membrane, right? Outer membrane. Very important, students. This, please make a note of all this. Okay, I will send you the notes on Telegram, but still I want all of you to make a note because these concepts are very important. If you do not understand where the site is, then for MCQ, you people will be really sad when MCQs come on the same. Okay. Now, students, site of oxygen, that is, where is your nascent oxygen present? Where is your nascent oxygen released? Okay, all the time you have to think about the image. Okay, all the time you have to think about the image. Tell me. Yes, lumen. Very good. Okay, lumen. It's in the lumen. Okay, next one more site. Okay, one more site. Site of your ATP and NADPH2. That means where is the site of ATP and NADPH2 getting synthesized? Where is all this happening? Very good. Stroma. It is happening in the stroma. I want all of you to answer. Okay, do not hesitate. All of you answer quickly. Students, even if you're first time to this class, no problem. Whatever you listened, just answer. Because when you type, what happens? Your brain also tries to read it, right? So subconsciously you are learning it with me. Okay, so yes. Okay, students, one more very important thing I already informed to you people is about the cytochrome, right? Cytochrome. So your cytochrome is actually a proton carrier. Okay, proton carrier. But also I can also call it, I can also call it as an electron. Okay, I can call it as an electron transporter. Do you agree? All of you agree to this? Do you all agree to this? Can I also call it an electron transporter? Yes or no? Yes or no? Yes. Okay. It is a proton carrier and it is also an electron transporter, right? Yes. Very good. Very good. So all of you understood chemiosmotic hypothesis. Did all of you understand chemiosmotic hypothesis? Yes or no? Yes or no? Tell me quickly. Okay. Let's look at this diagram and let me know quickly. All of you understood? Yes, I want all of you to understand. Isn't it an easy concept? Isn't it an easy concept? Yes. Oh, okay. Yes. Great. So we will do a very quick revision, okay? Quick revision for those who just joined. So what is going to happen? I'm going to just tell it in a few sentences, okay? Chemiosmotic hypothesis means the synthesis of your ATP. How does ADP gets converted to your ATP? Even though in non-cyclic photophosphorylation, we told that it is happening in the cytochrome, we did not tell how it is happening, okay? So chemiosmotic hypothesis helps us with synthesis of ATP, okay? Well, how does it happen? What happens is there will be a proton which will be accepted by the cytochrome. That proton will be sent into the lumen. Okay, now in the lumen photolysis is going to happen because during non-cyclic photophosphorylation we do not have an electron that is coming back, right? So because of that photolysis of water has to happen. Now during photolysis of water there is again protons which are formed and cytochrome also is accepting protons. So what is going to happen in the lumen? The proton accumulation is going to increase. It is going to be too high. So diffusion is going to happen. That means movement of protons from a region of higher concentration to a region of lower concentration. Now, where is proton at the lower concentration? It's in the stroma. Okay, so if the protons has to be moved from the lumen to the stroma, we need a protein. Okay, we need a protein. Students, I forgot to tell you, your F0 protein, right? Your F0 protein is an integral protein. That means it is present in between your plasma membranes. When you study plasma membrane, you would have studied integral proteins. That means proteins which are present in the middle, right? So F0 is an integral protein and we have F1 which acts as a ATPase. Okay. Now this proton is going to come out in a high energy, but when it crosses F0 and reaches F1, F1 will do synthesis of ATP by adding your phosphate to ADP and converting it into ATP. Yes, now the proton which has come out will go to your photosystem 1. After your ferredoxin accepts the electron, the proton will go to NADP, convert the NADP to NADPH2 with the help of NADP reductase. Yes. Isn't it clear? Isn't it clear? Yes, okay. Yes, CFO forms a transmembrane channel. That is actually an integral protein. When you study plasma membrane, you will study what is the transmembrane channel. Okay? Yes, very good. So all of you understood? All of you understood? Yes, yes. Super clear. You will never forget, no? You will never ever forget, right? Yes. Students, I also wanted to start your Calvin cycle. So I'll just give you an introduction. We will take it next class, but I will give you an introduction about your Calvin cycle. The problem is for Calvin cycle explanation, right? It's going to, it has to be broken down to many, states. So that's why I thought I will take it as an completely entire concept where I will take C3 and C4 so that we can do proper comparison. Okay, so now I will just introduce you. We will just go through what your NCRT is trying to say, and next class we will compare C3 and C4. Okay, all of you? Yes, yes, because I don't want chemiosmo hypothesis and your Calvin cycle to become, get mixed up. So we will go slowly to understand the cycles, because once we are done with the cycle, no, then this chapter is easy. right? Very easy. So students, Calvin cycle. I'll just give you an introduction about Calvin cycle. So what is happening during Calvin cycle? So Calvin cycle, the name itself comes from a scientist who figured this out, right? Most of this such kind of names are coming from the scientist itself. Yes. Yes, after this, you people have rapid fire, so do not forget. So be attentive. I'm going to ask you people question who are new to my class. Students, after every class, you're going to have a rapid fire. Yes? Yes? Okay. 1 minute. Yes. Okay. So, Calvin cycle was, yes, by Melvin Calvin. Okay. He was the person who found this out. Now, how did he find it out? He took the help of green algae. He took the help of green algae. What did he do with the green algae? He took the green algae. Okay. Yes. So imagine this is your green algae, students. He took the green algae and then he kept it, or he exposed it into sunlight. Okay. He exposed it to sunlight. Very, like, maximum sunlight. He kept exposing it. He was like, okay, let this algae get exposed. Okay. Now, this algae got exposed to sunlight. After that, he removed few cells. Okay, I'm drawing it here. He removed few cells of this green algae. Okay, he removed few cells of this green algae. So, I'll write here cells. Okay, and he tried to kill them. Okay, he killed them. Not only tried, he killed also successfully. Okay, he killed them with the help of hot. 1 minute. Yes, hot. Yes, yeah. He killed it, okay, killed them with the help of hot methanol. Okay, he killed it with the help of hot methanol. Okay, so he killed it. Okay, he killed it with hot methanol. What did he do? So for what was he trying to find? He was trying to find your traces, right? Students, normally I'll ask you this question, okay? Okay, I'll answer your questions. Okay, students, normally I'll ask you one question. You people have eaten, say, morning, you people happen to eat your, say, bread, or say your idli, dosa, whatever that you eat as a breakfast, right? Now, I'm a South Indian, so generally in South Indian homes, what happens? We will have idli, dosa, then upma, and sometimes poha, all this, right? These are the common food. Now, have you ever thought, how did you get the energy after after eating this food, right? Normally, if you're eating any food, how did you get the energy? How did you get the energy from it? That means there is some presence of a carbohydrate. That means there is some presence of your carbon compound, right, in the food that you eat. That is the reason why you're getting energy. Agreed? Agreed? All of you, have any of you thought, as soon as I eat food, how am I getting the energy? Yes or no? Tell me. Yes, ma'am. Yeah. So even we all have thought, right? Now, you have to understand that the carbon dioxide from the atmosphere, right? The carbon dioxide which is there, or the carbon dioxide which was there in the atmosphere, have to be accepted. In the, say, by the food, and that energy is what we are getting. All of you agree that we tell as carbohydrate, we tell glucose. Now, all of this is what carbon compounds, right? Carbon bonding has to happen. Now, where do we get generally carbons? We have to get it from the atmosphere. So, Melvin Calvin wanted to see the traces, right? He wanted to go behind and check what are the process that is happening? How am I getting energy? How is this, carbon dioxide entering the cell? This was his main thought. Okay, so he took C14, which is a radioactive isotope, and using C14, he did this whole experiment where he took a green algae, exposed it to sunlight, and then he took few cells out of it. He killed them with the help of methyl. Okay, all of you got this? And then what did he do? Why did he do all this? To check the products, to check what are the products that are evolved. That means after killing, what are the products that I'm getting, right? Now, normally what is happening? You take a plant, say now you're taking rice plant, right? What are you doing? We are taking it from the field. We are boiling it, we are cooking it, we are frying it, whatever we are doing with the rice, and we are eating it, but we are getting energy. So he did the same thing. He wanted to see in plants, what is that is giving us energy. So he did that. Okay, students, I'm just introducing you to Calvin cycle. Okay, now, so he was going behind the products. Now, when he was going behind the products, he found he found some of them which were, okay, some of them which were 3 carbon, some of them which were, okay, 5 carbon. Okay, he was trying to find that, okay, there were some 3 carbon compounds like your PGA. Okay, then there were like 5 carbon compounds, which is your RUBP. Okay, so he was like, okay, there are so many things. Now, which among this is a carbon acceptor? That means from the outside, the carbon has to come inside. Who is going to do the accepting? Okay, so let's see what your NCRT is trying to say. Okay, so the first, so your whole Calvin cycle, no, can be broken down into 3 main processes. I will get into the depth. Okay, I'll get into the depth later, but just try to understand carboxylation, reduction, and regeneration. Students, from the word carboxylation means what? Some carbon acceptance is happening, like some carbon is coming inside. So this carboxylation is basically fixation of your carbon dioxide. Okay, fixation of your carbon dioxide. Yes, fixation of your carbon dioxide. Very, very important. From the term itself, you can understand carboxylation, fixation of your carbon dioxide. And what happened? Carboxylation is the most crucial, your step of Calvin cycle. Why? Because we need energy. For that energy to come, we need carbon. Yes, agreed all of you? Yes, C6H12O6, your glucose. That is also again carbon. Okay, now this reaction, so what is happening? Carbon has to be accepted by some person. For this acceptance to happen, we need an enzyme. So now look here, where carbon dioxide is utilized for carboxylation of RUBP. Okay, RUBP. This reaction is catalyzed by an enzyme called your Rubisco. Students, where have we studied Rubisco, students? Rubisco. Rubisco, we have studied one place. Now, whoever tells me the answer, no, is my student, is a genuine student of my special class. We have studied Rubisco when we were talking something and we were like, oh, this is the most abundant. I'm giving you clues. Very good, Nandini. Excellent, excellent. Okay, biomolecules, right? Yes, Sanvi also, correct. When we studied biomolecules, I stressed on this in my special class, that Rubisco, right? Students, if you don't know about my special class, today I am coming up with Animal Kingdom one shot. Okay, I wanted to remind you people, now that we spoke about special class, let me tell you quickly. I know most of you are checking online what are the tricks to remember your Animal Kingdom examples. Students, I am doing today an whole one shot Animal Kingdom video where I will be making notes for each phylums with their characteristics, and also code for, or your phonics, like how Sonu eats sweets, right? Like that phonics for every the tricks are really funny, but then I'm going to make tricks for your whole Animal Kingdom. You can ask my students. Now we have already completed the chapter in depth, and now we, in the evening, we are going to have a quick revision. And I'm telling you, trust me on this, if you download that notes, it's more than enough for you to study Animal Kingdom, even if it's for your CBSE exam or even if it's for your NEET 2024. I can assure you, because I have made all codes for all the Animal Kingdom, including your Pisces classification. So if you still haven't followed me on my special class, students, you're losing upon so much amazing content. So go right now itself to the description box, click on the link, follow me so that you do not miss any notifications. Okay, and the special class is happening at 6 p.m. Okay, so all of you should be there. Will you all be there? Can I see new names today in my special class? Yes or no? Yes. Yes. Quickly, others also. Will you be all there in my special class, students? You just have to do one thing. You have to come to our app, download our app. If you already have the app, just search Gopika. Just search Gopika and you will get all my content. Yes, so I'm expecting all of you today in my special class. It's going to be a really fun session, students, because I have found such funny tricks to remember your Animal Kingdom. And if you're going to miss, then what do I say? Future doctors shouldn't miss any sessions, right? Okay, so let's get back to the class. So quickly, as soon as the live gets over, go to the description box and follow me, okay? Yes, so students, Rubisco. So your Rubisco is there. All of you know it is the most abundantly present protein. We studied it in our biomolecules. And then after that, okay, so what is happening? We are accepting carbon dioxide and we are going to get 2 molecules of your 3-PGA. Okay, do not worry. I will break it down very clearly in your next class. Just try to understand so much, because next class will be a little more easy. Okay, Now, your rubiscose is your carboxylase as well as your oxygenase. That means what? Whenever carbon dioxide is there, it will do that job. Whenever oxygen is there, it will do the job of adding oxygen. So it has a multi job, right? It will do both the job. Okay. Yes. No, the special class. Are you talking about animal kingdom, dear? Animal kingdom is happening in the special class. Animal kingdom is happening in the special class. Okay. Yes. So come and join our special class. You can download the app. The special class is completely free. Okay. Okay. Yes. Now, students, yes, it has more affinity to carbon dioxide, so it will always prefer carbon dioxide over oxygen. Now, second step, reduction. What does reduction means? Okay, reduction means, okay, so many doubts about the special class. Go to the description box, dear. Go to the description box, okay? Go to the special, download our app. Just click on the description box. There is a link for my special class. Join that special class and just type Gopika, or you can just go to any special class or just go to the app and type Gopika. You will get all my special class details, and today's I will also put it on the link, your Telegram. Okay, students, are you all on our Telegram channel, Unacademy NEET English channel? If you're not on our Unacademy NEET English channel, you are missing out on so many links and notes. Okay, yes. So students, reduction, very good. Addition of hydrogen, Sanvi, very good. Addition of hydrogen. Okay. So this addition of hydrogen, so this is a series of reactions that lead to the formation of a glucose. What are we trying to form? We are trying to form glucose. What are we trying to get? We are trying to get energy. Okay. Now this steps involves utilization of two molecules of ATP for phosphorylation. That means what? Addition of phosphate, and two molecules of NADPH for reduction. Now, per carbon dioxide molecules will require two ATPs and two NADPH. Okay. Now, the fixation of 6 molecules of carbon dioxide and 6 turns of cycle are required for the formation of 1 molecule of glucose. Do you understand why you should all eat food? Why you should all eat food? See how much effort it takes to make 1 molecule of glucose. Yes or no? Yes or no? Isn't it, students? Doesn't it take so much time to make glucose? And you people eat all this, junk food, and then we expect you to become what? To run like, to run in Olympics. Not happening, yeah. You people have to eat proper food. Got it? Yes. Now regeneration. Now what is going to happen? Your regeneration of your carbon dioxide acceptor molecule, that is your RUBP. Okay, RUBP is crucial if the cycle is to continue. That means what are they trying to say? Your RUBP should be regenerated. Otherwise, it is not going to accept the carbon dioxide. If it does not accept carbon dioxide, that means what? We are going to not form the glucose. So that means if carbon dioxide is not formed, that means energy also is not formed, right? So what are they trying to say? RUBP will accept the carbon dioxide, right? Will accept the carbon dioxide, convert it to glucose. But for this to happen, hero should be there. That is your RUBP. For that to happen, we need a process called regeneration. Okay? Got it? Now I'll show you one small image. Okay? One small image. Students, next class I will break it down. I will write a flowchart and give you. Do not worry. But right now, just understand what is this as a whole. Otherwise, it will be very difficult for you to understand C3 and C4 comparison. Okay? Yes. So here you look, students. We have our carbon dioxide, right? That is getting converted to our 3-PGA, which is 6 molecules. And then that is, for that to happen, for that PGA to get converted, for your G3P, we need 6 ATPs, right? 6 ATPs and 6 NADPH, that is for phosphorylation to happen. And then from there, we are forming our sugar, okay? Oh, sorry. Then we are forming our sugar, okay? Sugar formation is happening. Again, the cycle is going to continue. Why? Because we need to regenerate RUBP, okay? We are going to regenerate RUBP. Did you all understand the general idea of your Calvin cycle? So students, this is the easiest, okay? Look here. Chloroplast, you have your carbon dioxide. From your carbon dioxide, we take the carbon dioxide. In the Calvin cycle, we are going to end up forming your glucose. Okay? End up forming your glucose. Are we all clear? Can I get a good, put my favorite emoji. Come on. Quickly put my favorite emoji and we will end the session. And I will see you sharp at 6 o'clock for Animal Kingdom. Students, trust me, join and you will see how much fun classes are. Okay? And I'm going to give you a complete written notes from your phylum Porifera till still your mammals, I'm going to give you complete notes with their general characters, specialized characters, and your easy way to remember the trick. So only thing that you have to do is just click on the link. How is it so difficult? Just click on the link, right? Wow, most of my students know the emoji. I'm so happy. Yes, yes. Okay, great. So yes, 6 o'clock. Students, it's at 6 o'clock because the session is going to be long. I wanted to keep it a little early so all of you will be getting enough time to rest and have your dinner, because most of the time you people complain, no, ma'am, classes are too late. So I've kept it a little early. So quickly join the session. We will do a revision. If you people are not there, then there is no revision, right? Because you people are going to tell me the points and I'm going to write down the points. Okay? Yes. Students, do some adjustments and join the class, please. Okay? After you see what is your what is your priority, according to that, please do. Okay? Yes. So students, we have completed chemiosmotic hypothesis and we have got an introduction to Calvin cycle. Next class, I will do a mind map where I will draw and show you what is going to happen. Okay. So do not worry. We are going to understand each concepts. Even if it's going to take time, we are going to make sure to complete the concepts and complete the syllabus. Okay. And take part in the mock series so that you know where do you stand, because most of my students are really amazing. You people are answering also really well, so I expect all of you to check where do you stand in all India rank, right? So for that, only thing that you have to do is use my code. After using my code, you will get whatever discounts is required, right? All discounts you will get, as well as you'll also know where do you, like, you can just evaluate yourself. That's it. Yes. You people still didn't see my Malayalam shorts, sir, and you people are asking me, ma'am, are you a Mallu again? People didn't see me talking in Malayalam. Okay, let's do a quick rapid fire. We will do it in less than 2 minutes. Okay, very quick. Okay, are you all ready? Are you all ready? Yes, okay, I can see. But if you're ready, I want likes on the sessions. Okay, I don't want you people to just tell yes, ma'am. I want quick likes on the session. As the like increase, I'm going to see. Yes, quickly like the session. I'm waiting for the likes to increase. The moment likes increase, rapid fire is ready for you. Yes, like the session. Okay. So here is my rapid fire. Yes. Okay. Ready? So first question to all of you. What acts as an ATPase? What acts as an enzyme ATPase? What acts as an ATPase? Yes. Go on quick. First person's name I'm going to call. It's such a small name. So are you all sure? Are you all sure? Yes, Trisha got it right. Trisha got it right. Students, F0, that is your F0 is an integral protein. F1 is the only one that acts as an ATPase. Okay, so do not get it confused. Even though it is a full complex by itself, which will help the transport of protons, understand your F0 will allow the proton to come halfway. When it was about to reach your F1, the F1 will do the job of converting or forming ATP. So we will call ATPase as F1. All of you got it. Most of you have put F0. Did you have a confusion? Is the confusion clear? You can look into your NCRT also. You can see that the F1 is called as an ATPase. Are you all with me on this? Any doubts? Yes or no? Yes, cleared. Very good. So students, do not get confused. Okay, look at your NCRT. There is your F1 is the one that is called as your ATPase. Okay, next question quickly. Next question. When the proton is in the lumen, the stroma, stroma has high concentration of protein or low concentration of proton. Sorry, not protein, students, proton. High concentration of proton or less concentration of proton. When the proton is in the lumen. Very good. Nandini, correct. Low, right? Low. Okay. Next question. Are you all ready? Next question. Which is the most abundant protein that helps in Calvin cycle? The most abundant protein that is helping for Calvin cycle. Low concentration. Yes. Good job. Good job. All of you are answering. I can see all the names. I'm so happy. Yes, very good. The protein that is helping for accepting your carbon dioxide is your Rubisco. Very, very good. Rubisco. Yes. Students, the electron that is lost, by your photosystem 2 will be given back by who? Who will give back the electron that will be lost? Just tell me the molecule name. Just tell me the molecule name. Yes. Nandini is on fire. Full Josh. Okay. Before I could call anyone else's name, yes. Yes, what is uh Purple. Your name is Purple. Purple, if you want to join Avengers 3.0, the link is in the description. Just go click the link, enroll yourself, and use my code GOPIKA, where you will get a good amount of discount and you will get the whole package. That is your full 6 months physics, chemistry, and bio. For under 6 months, you will be getting from the top educators, and also you will get enough mock series, flashcards, and notes. Okay. Purple, just go to the description box, click on the link, type Gopika, and you will be able to join Avengers 3.0. You can just automatically click the link, Gopika code will automatically come. Yes? Just click the link and check if you're able to do it or not, or else you can join my special class and I'll tell you in detail how to go about it. Okay? But you can just click on the description box. Don't worry, it is going to open. And students, forgot to tell you, your Avengers 3.0 will get closed tomorrow. Okay, that means we will the session is going to start from 21st October. So if you still haven't enrolled, you are going to be late, because if you miss classes, then it is going to be really difficult for you to cope up. So please join before, by today night, make a decision and join Avengers 3.0. The link is in this video description. Okay, do not make it late. Even though we might be taking admissions, say we will be taking enrollments till 31st, we would expect all of you to be there when we launch the batch, right? So make the decision today night. Okay, that's the time given, so that you don't have to miss out. Like if you miss concepts, it's going to be difficult. Okay, all of you will make, so Purple, today is the right time for you to join, so quickly join. Okay. You can use Gopika code. Yeah, students. So the question was H2O, right? The answer is H2O. Very good. Next question. Next question. I'm thinking one question which I wanted to ask you people. Yeah, students, in Calvin cycle, what was which algae was taken? Which algae was taken? Very simple question, because in this questions are very less to ask. Students, join my Animal Kingdom. I'll give you a 15 rapid fire questions. I can ask you so many questions. Okay, in this questions are less. This is more conceptual, right? Very good. Green algae. Yes. Yes, definitely. Definitely you can crack NEET with your Avengers 3.0. It is a guarantee that we are giving you, or it is. Students, do not worry, because see, imagine 6 months time is there. Some of the students, even there are students who have worked hard, really hard for 4 months and cracked NEET. Okay, crack NEET. You can go check. But the thing is, you have to be consistent. You have to have the dedication. Only with that and with the teacher's help, you can crack. Okay, because see, students, YouTube is very distractive. So I always tell you people, join a batch where your teachers are guiding you. So Purple, you can crack NEET with Avengers 3.0. We assure you that, because 6 months your top educators are going to teach you, and they will make sure that every single concept is done in depth. Okay. Yes. Oh, that is very nice. Namaskaram. He's from Kerala. So yes. Okay, students. Thank you so much for joining. See you all at sharp 6 p.m. Animal Kingdom. Do not miss the session. If you want to crack NEET 2024, you know how much important Animal Kingdom chapter is, and you also know how important it is to remember all the examples, right? So do not forget to join. 6 p.m. I would expect all my YouTube students to be there, because I would love to see all your names in the new session, right? Yes, I have so many students from Kerala. Okay. More questions I will give you for animal kingdom students, because this has really less questions. This is more conceptual, so it does not make sense to ask question where is the proton going and all that. So for okay, from animal kingdom, I'll ask you minimum of 15 questions for rapid fire, and whoever answers this fast will be getting points. Okay? Yes. So can I expect all of you? Yes. Okay. Hello, hello. So class is getting over now. I'm telling hello. Students, join right now. Avengers 3.0, join right now and kickstart your NEET preparation, and let's crack it. Let's become future doctors, like I call you. Let all of my students be future doctors, so that next time when I see you all, you people will be like, ma'am, I used to watch your video. Right now, I'm a doctor. That is going to be the most proud moment, right? So, students, whenever you're lacking motivation, always come back to us. We are always here for you, right? We will give you the whatever that you need to crack 2024. But the only thing we expect you people to do is to download our apps, to be in the class consistently, and to follow us on a daily basis. Okay, that is all we are asking you for. The rest we will do it for you. Okay, you can trust us. Okay, you can trust us. Bye, students. See you all. Thank you so much for joining. Yes.